Alors donc aujourd'hui, je vais vous parler de ça. C'est l'amour de ma vie, T-cell antigen receptor. Du moment que j'ai participé à cette découverte importante au début des années 80. These are the people, the heroic people that actually have done the work that I'm going to present. The collaboration, this work on the T-cell antigen receptor was a great help from external collaborators in Leeds, before in Oxford, and then in Oxford. The Kennedy Institute, Michael Dustin and Stefan, and then Immunocore, there was also a collaboration. Then chemistry department in Rome, which was the university where I started. And then Omer, the dance. And I put Nicolas Destenville. You will see that Nicolas is in the audience, but he is the physique théorique. C'est la crème de la crème. And donc, I put it here actually, but I should have put it in another slide that is later. You will see it later. Okay, so the whole thing started with this, which I call it T-cell antigen evolution or revolution, which is the way. So in 1983 was discovered. In 85, this was the kind of dreaming about it. This was drawn, I think, in a congress together with Alice Reinert on a napkin. We're drinking something and say, okay, we should. A journal had asked us to give an interpretation of the T-cell receptor, and that was it was. And you can see the T alpha is alpha beta, but then becomes, about 10 years later, in the cloning era, an octamer forms of 4 dimers of dimers. Okay, so this is the alpha beta chain. So this is the one that recognizes really the antigen, or peptide antigen. You know, I'm sure most of the audience know about it. What is the ligand of the T-cell antigen receptor? And the other subunits which govern signal transduction, as people say. The most important thing is this immunoreceptor tyrosine based domain that was discovered soon, like in the late 80s. I'll tell you more, a bit in another slide. But this then in 2019 becomes this. This is a cryo-EM structure published in 2019. This is a detergent extracted T-cell antigen receptor. The subunits is alpha, beta, gamma, delta, epsilon, zeta. And in this structure, there is no, you don't have access to the intracellular tails because they are too mobile, you cannot see them. This was called by the Chinese group a 6 flavors ice cream. In fact, it looks like on the top, like an ice cream. This is the cone. You can see really the structure. So this is the cone. And maybe the tails are the little drops, the dropping of the cream, and this looks from the top like really 2 scoops of. Okay, so this is what looks a bit more realistically. This is a molecular dynamic simulation done in collaboration with Prakash, with Diraj, and Kali. These days you can, with appropriate people, you can model your receptor. This is a quaternary structure coming from the ER, from the cryo-EM. And then you can insert it in a bilayer, and this is an asymmetric bilayer. In this case, there's no lipids that carry negative charges, like the PEEPs, okay? And so if you wait and you observe this receptor, these tails are just always detached and like elongated, okay? Little secondary structure. You can see, I call it the octopus because in fact it's 8 octopus and there's 8 tentacles. You can make the octopus dancing if you want. So by doing a simulation, and you can see that this is about 20 microseconds. of observation. And now, what they have done in this simulation, they have added on the inner leaflet some pips. And the pips actually attract the tails through the negative charges, the positive charges there are many positive charges in these tails, and some of the tyrosine actually are hidden inside. So they really energetically have favored the retraction of the tails, and also the tyrosines. Not all actually, but some, at least in all the snapshots that one can look at this, because that's is many. It's like looking at the movie, one after the other, and you can stop on one, and this is one of the negatives, essentially. But if you then arrive at the end of the simulation, which is a bit longer than 20 microseconds, you can see one thing which is quite interesting, is that the tails are retracted, but actually in different simulations, or at least where they stopped, okay? Of course, it is a random event, but they get to the point of all the tails which are attached to the membrane, and they in different configurations, okay, which is quite interesting. And also, we notice also that you can see there look at, this is the alpha beta. Sometimes it's in this position, sometimes more, so essentially the receptor is moving. Also the ectodomains, if you look carefully, that you see in yellow, so the ectodomains is this part of the receptor outside, okay, and also in this part, the CD3 subunits and the constant regions of the alpha beta, and also they actually, for each configuration, they're slightly different, okay. So maybe you're thinking that there is a certain coupling, perhaps, between what happens in the ectodomains, what happens in the transmembrane regions, and what happens in the tails, okay. This coupling, they looked, our collaborators actually, they looked carefully at this. The coupling is not perfect. So there's some stochastic events, but actually there look to be some deterministic facts that, in fact, that a certain type of configurations of the ectodomain, there is also a make you thinking already about a potential model for an allosteric regulation of the receptor. Something changing outside can correspond to some changes inside. Okay, no, the mystery of this receptor is its complexity, as you can see, and there's no associated tyrosine kinase. It's like a ghost, but upon binding, so essentially biochemically, you cannot really demonstrate unless you really force the sensitivity. Probably to the point of an artifact, you don't see really an associated tyrosine kinase, protein tyrosine kinase. Yet, as soon as you add peptide MHC, so something that binds here, in a second, you can see already phosphorylation of the atoms. So it has to be there, but consider a second is a long time. It's not, you say, as a biologist, we think that 1 second is really short. But in terms of this type of mechanism, it's probably, I wouldn't say biblical time. Oh, it is biblical time, yes. So it's really centuries, so order of magnitude to events to take place. Okay, then there is another kinase called ZAP70 that binds. And what this series of events in cascade lead to the activation of the so-called signalisome, and then T-cell activation begins. Now, for a number of years, especially in Oxford, I became very interested in trying to clarify, through support with Wellcome Trust, to try, I lost it. You don't see the, of course, yeah. Interested in these two aspects, how the binding activates the kinase, if any, and how it exposes perhaps the tails to be phosphorylated by LCK. This is just the beginning of how things work. If you don't do that, you don't activate a lymphocyte. You don't have activation of a lymphocyte. And thanks to this work that we published in 2010, the first name was Costantina Nica. Oh, it's very difficult to, I have to concentrate here, because otherwise, yeah. Costantina. Essentially, what we
published there what could be considered an anathema, essentially that, that LCK was already activated. There was about 50% of LCK, maybe 35, 40% in normal T cells. This is already active. So we made a lot of controls to make sure, in any way and shape, to make sure that this was not an artifact, and this has been confirmed by others, with other means, other approaches. But, so LCK is a Src kinase, and when you say a Src kinase activated, people say, oh, cancer. It's not true. It is regulated, and I will show at the end a little bit of work that we have done to define a bit better this, and also some unexpected properties of how LCK is maintained in the membrane, which led us to collaborate with Nicolas here in Toulouse. Okay. It's considerably active. So, okay, so what we thought very simply is that it's active. So the only thing that the receptor has to do, you can have other models, you can think about it, but we thought we'd like to think simple. So that, in fact, the only thing you need is that the receptor, it's dangerous to think very simplistically, of course, but you know, there are some plausibility of that you can relate to other observations. And so this idea, it doesn't come out of the blue, but it actually is based on observations of others as well, which point to a possibility of this mechanism. So you can see this means a conformational change somehow that has now allowed the famous tails of the octopus coming out. And now LCKD is active, is there, bang. So it can be there, but you don't see it. Mostly because these tyrosines are hidden or protected in a way. Okay, and then, well, I just want to say for the aficionados, immunologists of CD4 and CD8, that it is known since a long, long time, since my time I was in Lausanne, in fact, was discovered there, that the co-receptors, especially CD8 was studied there, are not really needed for I call it the butler of T-cell receptor. In other words, yeah, they actually very active and they contribute when something is missing. You know, the butler in a college is somebody that you need some wine, and the guy immediately goes and then brings, oh, professor, yes, okay. This servile type of things that I don't like very much, but it is like that. And so, but it's there essentially to help the receptor, also to reduce the off rate of peptide MHC binding to the TCR. That's the main function. Okay, the reason, and then we thought about, oh, this could be an allosteric activation, but we were against a number when I had some ideas about this thing, but not necessarily was convinced about it, but that there was a whole series of negative data in the literature. People were expecting, by looking at the first structures that started in 1998, I think, Chris Garcia and Don Wally, they published these first structures of TCR complexes with peptide MHC. And so in some cases, people were obtaining a structure of unbound and bound receptor. And they looked for if there was any change that in structure upon binding that was behind the variable regions. In other words, if there was any sense that there was some transmission of conformational change to the constant region. So this is the primordial idea for a possible allosteric mechanism. And so in reviews, people say, no, this is not possible, and blah, 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 blah. Okay. But of course, okay, let's forget about other comments. But there was one exception that was published in 2003 and then comforted by another publication in 2009 by a group in Australia, where they actually, in crystallographic structure, in one out of already some complexes, could see some changes in a crystal. And then starting 2016, 17, this bunch of people they actually start to publish NMR data. So you purify, you have alpha, beta in solution, you add the ligand, and then you can, by labeling, you can do, you can see how the atoms, the side chains and the main chain is moving. Okay, and you can make correlations. This is very, very powerful, and that's the best way to look at conformation, at allosteric mechanisms. Okay, unfortunately, you cannot do it with large proteins. But, so what they concluded was that there were allosteric sites present in a constant region upon binding. In other words, you bind and you see some movement, something happening at distance. And those sites where it was the first to show this was Roy Mariuzza and Orban, were coinciding exactly with sites that interact with the CD3 subunits. But why cannot go somewhere else? Why they should aboutir là-bas? Why they should arrive there? Exactly where the interaction with the CD3 subunits is. And so at that time, the model proposed to explain how the T-cell receptor was starting from binding to activation were two types of models. They were fighting with each other, and blah, blah, blah. And so one is a conformational change independent, which I call a rigid body model. So essentially, the T-cell receptor is a bit stupid. It doesn't do anything. In fact, in all cases, it's a bit stupid. It doesn't do anything, and it's actually dependent for seemingly on other events. It cannot reason itself. It's not constructed, built up in a way to induce the activation of itself, which is an important concept. And so some people thought about that the CD45, this phosphatase is taken away, and this is LCK is activated. We know that LCK is already active. Or co-receptors or receptor clustering. And they're all, you can, if you think about well and you look at the literature data, you actually, because you have always to look at the literature, what the literature says, okay, and confront with whatever idea comes up. And in fact, there were very critical points that were not explained by this idea. There was a second one, mechanosensing, came very much in vogue. Now, everybody likes mechano. A lot of physicists, they come into work, and they, of course, they have to find a job. I'm sorry, but it's like that. So I like very much to put force everywhere. Of course, force is everywhere. Okay, there are some forces, in fact, when the TCR engages with hepatitis C. But how frequent they are, how oriented they are, it's stochastic, essentially, mostly. It's difficult to imagine that it's always the same. Okay, and so. We can discuss about it. And the other was a pervasive geometry, which essentially required that pre-assembled peptide MHC together, clusters, which is also something which is not, for a number of reasons, is quite a problem. However, a genuine allosteric mechanism, I call it, the Monod-Wyman-Chanjou model, nobody considered it. It was something like, like anathema was, no. In fact, I mean, it was like that. So it was like shine, so hate and fear, kind of, no, it's not possible, which is a typical thing of occupying sites in the publication and then not wanted other ideas to come in. Unfortunately, it is like that. But it is like that. Okay, so, so the idea that I had at that time, so it was about 8 years ago, was to say, okay, can we do a genetic screen by adding mutation in certain areas of the receptor which may be involved in this mechanism? If you look at the structure, the quaternary structure was not known at that time. So it's sort of imagination of a poor biologist. They say, okay, if I had nothing, I do genetics. I start to perturb and then see what happens. And the idea was quite simple. In fact, I was trained in genetics by Roberto Tosi when I did my thesis. So essentially, thing comes always good at a certain point. And so the idea was that if the transmembrane domains, or regions, these alpha helices, are really connect the external part to the internal part, the question is, if I'm going to this is the part that should ensure somehow, of course there are iuct-o domains that we have seen, but this should be perturbed during a potential signal transmission, and in a case of an allosteric model. And this is where the signal, in a way, has to transit. Okay? It's a physical thing. And so the idea was very simple. Say, okay, if we, and so if these regions, transmembrane in particular, are just needed for receptor solvation in the lipid bilayer. It's a physical thing, so it has to be, okay, a basta. Okay, doesn't do anything, this is Italian, okay? Okay, and then, are important for the topology of the complex, but why it's so complicated, by the way. Then, if I do mutations there, it was a bet. Probably was not the best tight type of experiment, but that's the best we could think at that time. So if you do mutations there and we don't see changes in intrinsic capacity of the receptor to signal, okay? We haven't done anything. There are experimental criteria that you can use very clearly to check that. Then means that really that hypothesis is correct. But if we are seeing something different, maybe a decrease or say, we were expecting a decrease in signaling. You touch something there and you have an impediment of signaling. So I found the good, and then we use, of course, a system with antigen specific T-cell receptor that we can stimulate, in this case by tetramers at that time. And I found the German postdoc in Freiburg in a meeting, and I told him, he looked very enthusiastic. I said, oh yeah, why don't you come to the lab and as a postdoc and you do the systematic mutations of all the subunits of the T-cell receptor. I said, oh yeah, sure, I'm coming. Sure. He was clear. So I said, okay. And this was drinking a coffee. I like, best ideas come when you drink a coffee. You sit down, relax, you drink a coffee, and then, oh yeah, we should do this. Okay, and then you think about, no, we shouldn't do it. Okay, so essentially, what was observed in these experiments, this is a systematic mutation. This is an RNA scanning, or it's called leucine scanning. So essentially, what it took the beta chain. We started with the beta chain. You wanted to do everything. Then I had to stop him because no, we don't have to go to all the T-cell receptors. There was already enough information that we can actually, this is very interesting. Okay, so you notice that what was detected here, I passed on the experimental setting for this, but that was when you introduce certain mutations, you get a decrease in expression of the receptor. You perturb the structure so much that quality controls in the cell tend to put in a bin. Okay, let's say eliminate. They cannot go to the membrane. Some mutations, there are more than this. This is just the most representative and most studied. Some have no effect. Some others, like this one, this is very, very important. You mutate this lysine in the beta chain, and no more T-cell receptor on the surface. Bang! Everything is one single amino acid wipes out. We did it in 1990. We published a paper on that with this idea, but it was only 20 years before, so it was not good time. But what we noticed that was in the particular residues in this area here, these three residues, in particular this tyrosine mutated into alanine, produced an increase in response. This was a very crude assay initially, but it was repeated so many times. I told the postdoc when he came once to the data, I said, no, you go back, please, and you repeated this at least 9 times. And then if you find it, it's okay, and with good controls. And he said, yeah, sure, I'll do it. He did it actually, but maybe even more. And so, and actually, it came out very significant that there was an increase in signal. Now, increase in signal means in this case that the T cell has been expressing this receptor. This is a T cell line, convenient for this type of experiments. And then you stimulate with a peptide MHC in the form of tetramer, and you can see there is an increase in signal, despite the fact that the receptor at the surface even decreased. This was really paradoxical. Okay, so we thought, well, now we have really to understand what's going on. And so to solve this enigma, we had to do a couple of new things. First, to have a very accurate signaling assay, okay? So to compare wild type and mutant may be in parallel by barcoding, by fax analysis, because ERK activation of Z phosphozeta phosphorylation, zeta phosphorylation can be also detected very carefully by and quantitatively by fax analysis, and then also a biochemical assay that will tell us what was going on in the receptor when this mutation was, and finally with atomistic initially, and then coarse-grained molecular dynamic simulations. The first thing was very nice, so that you can see this is a dose response using tetramers at these concentrations of a receptor which has an affinity of, in this case, of about, I think is 16 or 18 micromolar. Okay, you can stimulate, and you can see that the mutant actually gives not really a change in IC50, okay, EC50, I'm sorry, which means that the affinity has not changed. The mutation in the transmembrane has not really changed something in terms of capacity of recognition, of which you may think about if it's a possibility, but actually was more efficient. When you have this type of graph, you go to Omer Duszek in our department and say, Omer, can you understand? Of course, we understood because he had published something to indicate that, in fact, if you consider that your receptor is operating in a regime of proofreading, kinetic proofreading, that's the kind of expectation. I'm not going into details, but essentially the data were plotted now using this expression, in which the Kp can be plotted here, and you can see that in fact there is an increase in Kp. The Kp is an intrinsic forward is a kinetic parameter that tells you that the signaling is more efficient, is faster, okay? Because it probably happens in better conditions. So we found that other mutations also that did not affect surface expression didn't show any gain of function. This is a gain of function mutation, okay? And also we found other mutations. Of course, we are not going to show to you, but there were other mutations around the tyrosine. Actually, were increasing, and also we extended to other receptors because a reviewer said that because we had worked only on a receptor which was anti-tumor receptor, we had to show it also for other receptors which were antiviral receptors. So believing that they could behave differently, which for us was a bit strange. But that actually was a good suggestion because we had to move to another system, an anti-HIV receptor also, against an antigen of, which I forgot the name now, and then to try to show if these things were true, and they were true also for other receptors. So this augmented signaling was not caused by all those things that you can see there, and I did go very, very fast. But the reduced expression, we reasoned that the most likely reason that the reason was most likely was because of instability of the receptor, as I was telling you at the beginning. So this reduction was by 20%, 30% maximum of, you can see very well in fax analysis, especially if you overlap the two wild type and mutant, and you run fax, so you induce the lines because these are receptors that are induced at the same time. So it's all technical aspects here, which are very important, so tedious, but at the end, you gain in terms of robustness of the data and reproducibility. Okay, so instability. So that one expectation was that if there was some instability in the complex induced by the mutation, you would expect, and that's work that I've done, actually, it was the first paper that we published from Paris, actually, that it could mean that, in fact, the mutation is somehow detaching a bit the subunits,
And if you extract the receptor now with a mild detergent, you can see that the stoichiometry of the receptor is decreased. So epsilon, gamma, or zeta could be not present with the TCR with the same stoichiometry as wild type. Okay? You scratch, detergent extraction is nothing but scratching lipids in the complex or around the receptor, and then substituting some with detergent molecules. And what you do is, you can, by doing that, you can lower the lateral interaction in the transmembrane region. Okay. A little bit of biophysics, or say biochemistry, which is a good thing to know. Okay, so then we started collaborating with Diraj and Andreas in Leeds, and so they actually look into the atomic coordinates in the cryo-EM structure, and they found that this tyrosine was in a particular critical position that actually interacted with at least 3 residues. They seem distant here, but in space actually they're quite close to each other. And then the possibility was that the substitution of this tyrosine with a small side chain like alanine could be actually creating space and inducing some movements. And in fact, what they found, they did quite a number of simulations, and they found that the angle between the gamma epsilon and alpha beta. Gamma epsilon is the one that really is close to this tyrosine. If I'm the alpha beta, I'm the beta, and the gamma epsilon is here. Okay, it's close. The epsilon delta is almost on the other side, close to the alpha. In fact, the angle was quite changed. So it means a movement. And so that, in fact, that's, as I say, this tyrosine was making contacts with quite a number of, and then its mutation would imply some important movements in the transmembrane regions, and another configuration. So then we have to go to some experimental demonstration, and as I anticipated before, this was based by this assay, we call it DSA, or DDM stability assay. DDM is the name of the detergent used, or lauryl maltoside. We call it lauryl maltoside, and that's when you take the wild type receptor and you pull it down. We put a tag here inside, and you pull it down, and then you look at and you quantitate very carefully the subunits by Western blot, believe it or not. Then you have, you can see the beta chain. This beta 2 is this one at the membrane. Okay, the other two, actually, this beta 1 and beta 3 actually are forms of beta that are inside the cytoplasm. Those are details again, but essentially by looking, for instance, at the ratio between zeta and beta 2, or epsilon and all the other subunits, you actually, the sum of the other subunits, essentially you establish a stoichiometry of all the zeta, epsilon, delta, and gamma with respect to the rest of the complex, okay? So if you do that, and now the answer is that if we now go into the mutant, what happens? The mutant, this showed exactly what's predicted. So that if you look at what the mutation does in terms of recovery of zeta here, or epsilon, in fact, is decreased. I added a sentence here to say that this is not possible that those are detached already at the plasma membrane, because otherwise the receptor will never go to the surface. Okay, so in fact, the receptor is intact at the surface, okay, with some decrease in expression, in number of molecules present on the surface. Also, gamma and delta were actually slightly decreased. So the receptor has actually loosened it, okay? And the detergent make appearing this. Now, I go very fast here. What we wanted to prove ourselves wrong. It's a good thing to do. It's a bit masochistic, but it's okay. You can do it as a scientist. Scientists are a bit strange people. We know we are strange people. Okay, so
yeah, a bit masochistic. And then you try to prove whether the mutations now in the zeta chain, okay, can also, some mutation in the zeta chain can also induce this loosening and result in a gain of function. Well, the answer is yes. And I skip all the slides, 3 slides, to show you this, okay? It's all published anywhere, okay? So they identify 2 residues, which were important for zeta to be packed against the alpha and the beta. And when you, because they interact like this against the alpha beta, okay? And so, and they make actually contacts with many other, the zeta makes many contacts. Okay, and of course I forgot to have the time. Sorry. Anyway, so the answer was that the mutant in a simulation, which is shown here, in fact moves more. You can see, you can actually see this behavior. Okay, so it was these mutations were loosening zeta vis-a-vis the rest of the complex, and in fact gain of function and reduced expression and blah, blah, blah. So the conclusion from all this tedious, long analysis to be sure that we're not falling into a well, a black hole, was to say that single mutations in transmembrane regions can cause some loosening or relaxation of the structure, which results in a gain of function. Okay? The other details which I spare, but only in the paper, that actually comfort this idea with other evidences, like the receptor with this mutation start signaling already at basal level, already like is frying in terms of signaling. You can see some basal signaling, which is then amplified by the ligand. Now, the question was then, is the mutation effect mimicked by the ligand? And the answer is yes. There was really an explosive champagne in the lab because it was correct. So we did another type of assay similar to the previous one, and was essentially to do this. You add the ligand, and you use receptors which have higher affinity than normal, or they are in the spectrum of affinity, which is about 100 nanomolar KD or 50 or 20. They exist. Some CTL clones, memory cells, highly selected, they have affinities which go to 50 nanomolar, okay, quite high. When you do this, you can take you can bind your ligand, then you solubilize the cell very, very quickly for 4 degrees. Then you capture the ligand. This case was tetramers. And then you look at the state of the receptor compared to a receptor which is not being engaged. You can compare the stoichiometry of these two, an engaged receptor with a non-engaged receptor. And the answer is that, in fact, you can see that there's many, many other experiments that, in fact, these are two receptors. This is an anti-HIV, and this is, and add the name, and I forgot the name, of course. It's a peptide antigen, of course, from HIV, but okay. And then I should look in the paper. And another one, this is about 50 nanomolar, and this one is 140 nanomolar. Okay, okay. This is natural. This has been affinity matured. Well, you can see clearly, and just looking at the zeta, that in fact, if you look at the status of the ratio between zeta and beta, okay, you can see that there is less zeta. Okay, that means that something has happened, the ligand has done something. Looks like a crude assay, but actually allows you to measure and very, very reproducible. This was also 4 degrees, which comforted that this could be really a conformational change. And also in intact cells, but also in solubilized TCR. You take solubilized TCR with this detergent, you add the ligand, then you capture the non-engaged or engaged, and then you show that, in fact, also there is zeta, you recover less zeta, okay? Then we went a little bit ahead, and we started to think of another possibility Okay, now we engage the receptor with a tetramer. What if we engage the receptor with a monomer? This was anathema, once again. So a monomer engaging the receptor and is causing, but if this was true, this was exactly a demonstration of an allosteric effect. Okay. Purists would say, yeah, but you should look exactly there. Yeah, but if I show formally that upon binding, I have an effect of this type, well, it is not the Holy Spirit. If you want to believe in the Holy Spirit, you're free to do it, but I don't. So there must be something happening. And in general, these are clearly allosteric effects. Ligand monomeric, monodispersed, added, and what happens? To our surprise, no, it's not a surprise, we like that actually, that in fact also there was a clear decrease in zeta, but also the other thing was that this was another thing that people say, oh, that's really strange, do you find this thing? That in fact the monomer is able, because of this affinity, to stimulate, you can see ERK activation, the signal is going through the membrane, it works, okay? And last night at dinner I asked Salvatore, who can do a single cell calcium analysis of this? I said, no, he said, no, me, not me. My lab is full of people, people asking. So I have to find someone maybe in Paris, or maybe you'll do it one day, if I have convinced you, which is worth it, without the data that I'm not going to show today. And so, and other receptors, this is, for instance, okay, sorry. Yeah, okay, so well, the same thing happened with, these are two different receptors, the usual that we have used most, 50 and 20 nanomolar affinity. The reason for this high affinity is that it doesn't change. We have the structure of this. The engagement of the receptor is canonical. There's nothing strange about it, okay? So, and then also in T cells, I'm sorry, there's been something. This is not an I, it's a T. So this receptor was transduced into T cells, and then we checked. In fact, look at the nice. This is primary T cells, of course, memory, and look how nicely they're stimulated in ERK when you add the ligand. We did also some controls of this is a monodisperse. So you check actually that the ligand is passed through a gel filtration, and you can see monodispersed, no problem. So there's no aggregation. The other thing is also there could be the possibility that, which I thought at night, there was a nightmare for me, thinking that you add the ligand binds to the cells a little bit, and then they can stimulate in trans as from a membrane. And in fact, we did all kinds of controls that was not the case. This is a crazy thing that you can think about at night and say, and you don't sleep at night, say, what kind of experiment should I do? Okay, and this is also solubilized TCR. I mean, so essentially the monomer does work and does induce a signal and a conformational change detected with DSA. So we arrived to this conclusion that in an unliganded receptor, the configuration of the CD3 subunit has to be what you see there, the first one. In the liganded, maybe we like the idea that the lipids actually are moving and then creating a situation that's in line a little bit with what Wolfgang Schamel has published about the possibility that also Mark Davis, I think, published a bit of a lousy paper, actually. We don't like it very much, but okay, published papers saying that cholesterol would probably, if you subtract cholesterol, the T cell receptor actually becomes a bit change and can actually signal better. In the latest experiments that they published, a group in China actually, they've found this. So, corollary and remarks about this. The allosteric regulation in this case is probably what we call, it can be called entropic allostery. So it is in fact something that is very hard to see by, and its existence has been documented, no problem. You cannot really see very easily in crystal structures, which are energy minima, so very, okay, and very stable. There are simply changes in dynamics that are like a wave, if you can visualize in this way, is repercuted at distance. So it's just mostly an entropic effect. The other thing is that what is an interesting idea about the allosteric regulation is that let's say conformational change travel at a speed of 1 nanometer per second, per microsecond. So in a few microseconds, upon binding, you get an effect. So people have measured this in various proteins, in various, even dimeric proteins that are allosterically regulated, and they could register by NMR or other means that the time that takes, mostly NMR, from the time you bind to the moment you see changes at distance, but a long distance in the protein, maybe in a catalytic binding site, it takes about 1 millisecond. Okay, so we know one thing, that binding of even binding of the weakest receptors to the usually are self-peptide MHC last, has been measured in various labs, in particular by Cheng Zhu in Atlanta. It could last, so the minimum is 100 milliseconds. Now, if a conformational change takes 1-2 milliseconds to go from one side to another, 100 milliseconds is a lot. So it becomes almost independent of time. So the time, these 100 milliseconds are important for accomplishing phosphorylations and other things that has to happen inside. I think this is an important point that has been completely in the oblivion for people that work in the field. The other thing is that probably we would like to think that allosteric activation, now think about also the NMR data of other people, that also new data that just came out a couple of months ago that agree with us, that in fact could be the beginning of a cascade of events. First, conformational change that travels very fast, but there are also co-receptors that can come. It is known that co-receptors are approaching signaling TCRs. So there is something that is really a bit magic and probably understandable in certain terms, that the co-receptor comes only when the receptor has been engaged and is signaling, which makes sense. The exclusion of phosphatases and catch bonding and other things could be also seen, all these things, as a positive feedback and amplification of the signal, a stabilization against what I would call a chemical and mechanical noise. A T-cell receptor is living in a membrane which is very populated by the proteins. It's completely, totally disturbed. Okay, and so maybe I spare you this, just to tell you that just the results instead of showing you all the slides. That it is known, just a concept, that the T-cell receptor can be oriented on peptide MHC preferentially in a diagonal configuration. If I may show you, I don't know if the camera is showing, if this is peptide MHC, it has to be diagonal, okay? And it makes some sense from a structural point of view and so on. And so this is an illustration of a paper where the plotted, the angles, the crossing angle is exactly what I'm telling you, okay? And the tilting angle is like the receptor can, in fact, by having all these movements across peptide MHC, it's, and changes in configuration slightly, the diagonal orientation maximizes the TCR view or readout of peptide antigen. That's what it does. Okay, it lands on the MHC, but actually at the end has to recognize and increase its affinity by looking at the peptide. Otherwise, it will not work. So we are quite lucky to have structures of 2 receptors with a company in Oxford. Very lucky that have canonical or non-canonical orientation. So in this case is exactly a receptor. It's about 180 degrees in a diagonal orientation vis-a-vis peptide MHC. It binds with a good affinity, you can see. It's an affinity of 3.2 micromolar. The conventional receptor binds with 36, about 10. Now, if we look at how they respond, well, big surprise. Only the canonical signals, and the other doesn't, despite the fact that it is actually even higher affinity. This is tetramer binding. That shows also on the cells that there is, in fact, about 10-fold difference in binding. And you can see that the receptor, which is oriented just 180 degrees on peptide MHC, is not working. You can increase peptide. There's 3 types of responses. The first one is phosphor-ERK. This one is CD69 expression. This is one is gamma response. This is a reconstituted T cell with these 2 receptors. So the non-canonical doesn't work. Okay, so I will spare you really because we have done also molecular dynamic simulations with a group in Rome, theoretical chemists that do this for us, and they come out with the fact that, in fact, the conformational trajectories that you can see with non-conventional receptor oriented in 180 degrees, they're very, very different, and some have disappeared. Meaning that probably the conformational change, which starts from the alpha V beta binding and then is transmitted to the constant regions, mostly beta, we know that, initially is not the same. And maybe this means that something cannot happen downstream. Okay, so this is the analysis that has been done. So essentially, the deorientation is required, the canonical orientation is required for inducing optimal signaling. Believe it or not, but the TCRs which can bind orthogonally cannot signal, and some that bind parallel on the peptide MHC cannot signal as well. Okay, so it's quite interesting because they've never been seen. The structure has never been seen. That means they cannot be selected in the times. Now, alpha beta signal depends on 2 main parameters. So this is the conclusion that we have. It's allosterically induced conformational changes and binding kinetics. And also that we think that this is a demonstration, also this particular last cherry on the cake, that you can have binding with good affinity, but no signal. It means that there must be an allosteric effect, a conformational change that is important in order to then allow something to happen. And the kinetics is the time required for the time that you have to give to the receptor to phosphorylate and having all kind of number of events that so signal is full, is exploding really, okay? I think I have no time, unfortunately, and I'm really sorry because I have to, can you invite me next time so I can come and, okay. I can come for, even for students, I'll be very, very, but essentially, I think I'll stop here because otherwise I'm already taking much of the time. And, but Salvatore told me to be as simple as possible. I hope, I really hope that you guys have followed with patience. And I promise, actually, why don't you invite me? I come to, and I show this. It's very close. I mean, it's 4 hours from Paris. It's very, very easy. Thank you very much. Thank you for your attention. When the vision's there.